Okay, 14.3 is studying the human genome. So, just a few decades ago, computers were gigantic machines found only in laboratories and universities, and today many of us carry around small, powerful computers to school and work every day. So, decades ago, the human genome was unknown, and today we can see our entire genome on the internet. So, how long will it be before having a copy of your own genome is as ordinary as carrying around a cell phone or something else in your pocket? So, what techniques are used to study human DNA? Tools that cut, separate, and then replicate DNA base by base. Scientists can now read the base sequences in DNA from any cell. So DNA is a huge molecule. Even the smallest human chromosome contains only 50 million base pairs. Manipulating such a large molecule is extremely difficult. In the 1970s, scientists found that they could use natural enzymes in DNA analysis. Scientists can now read the base sequences of DNA by using these enzymes to cut, separate, and replicate DNA base by base. Cutting DNA. Nucleic acids are chemically different from other macromolecules such as proteins and carbohydrates. This difference makes DNA relatively easy to extract from cells and tissues. DNA molecules from most organisms are much too large to be analyzed, so they must first be cut into smaller pieces. Many bacteria produce restriction enzymes that cut DNA molecules into precise pieces called restriction fragments that are several hundred bases in length. Of the hundreds of known restriction enzymes, each cuts DNA at a different sequence of nucleotides. And there are some examples of cutting DNA that are in your book, and it will be easier if you just look at the pictures there to see how that happens. And pay attention when they talk about leaving single-stranded base overhangs and sticky ends with the sequence of AATT, because the sticky ends bond or stick to the DNA fragments with the complementary base sequence. Separating DNA. Once DNA has been cut by restriction enzymes, scientists can use a technique known as gel electrophoresis to separate and analyze the different sized fragments. And separating DNA. A mixture of DNA fragments is placed at one end of a porous gel, and when the electric voltage is applied to the gel, DNA molecules, which are negatively charged, move towards the positive end of the gel. The smaller the DNA fragment, the faster and further that it will move. So, separating DNA, the result is a pattern of bands based on fragment size. Specific stain that bind to DNA makes these bands visible. And researchers can remove individual restriction fragments from the gel and study them later. So after the DNA fragments have been separated, researchers can read and sequence it. So single-stranded DNA is placed in a test tube containing DNA polymerase, the enzyme that copies DNA, along with the 4 nucleotide bases, A, T, C, and G. The DNA polymerase uses the unknown strands as a template to make one new DNA strand after another. So researchers also add a number of bases that have a chemical dye attached. And each time a dye-labeled base is added to a new DNA strand, the synthesis of that strand stops. And when DNA synthesis is completed, the result is a series of color-coded DNA fragments at different lengths. So the researchers then separate these fragments, often by gel electrophoresis. And the order of color bands on the gel tells the exact sequence of bases in the DNA. and the entire process can be automated and controlled by computers so that DNA sequencing machines can read thousands of bases in just a matter of seconds. So, what were the goals of the Human Genome Project, and what have we learned so far? Well, the Human Genome Project was a 13-year international effort with the main goal of sequencing all 3 billion pairs of human DNA and identifying all human genes. The Human Genome Project pinpointed genes and associated particular sequences in those genes with numerous diseases and disorders. It also identified about 3 million locations where single-base DNA differences occurred in humans. So in 1990, the United States, along with several other countries, launched the Human Genome Project. The main goal of the project were to sequence all 3 billion base pairs of human DNA and identify all the human genes. Other important goals included sequencing the genomes of model organisms to interpret human DNA, developing technology to support the research, exploring gene functions, studying human variation, and training future scientists. So, how can the huge amount of DNA in the human genome be sequenced quickly? Well, researchers first break up the entire genome into manageable pieces. The base sequences in widely separated regions of a DNA strand are determined. These regions can then be used as markers, and the markers make it possible for researchers to locate and return to specific locations in DNA. So once researchers have marked the DNA strands, they can use a shotgun sequencing, a rapid sequencing method that involves cutting DNA into random segments, random fragments, then determining the base sequence in each fragment. Computer programs take the sequencing data, they find areas of overlap between the fragments, and put the fragments together by linking the overlapping areas. The computers then align those fragments relative to the known markers on each chromosome. So much of today's research explores the data from the Human Genome Project to look for genes in the DNA sequences that control them. By locating sequences known to be promoters, binding sites for RNA polymerase, scientists can identify many different genes. So shortly after a promoter, there's usually an area called an open reading frame, which is a sequence of DNA bases that will produce a messenger RNA sequence. And once other sites that help to identify genes are sequences that separate introns from exons and stop codons located at the ends of other open reading frames. So if you were to compare the genomes of two unrelated individuals, you would find that most, but not all, of their DNA matches base for base with each other. So on average, one base in 1,200 will not match between two individuals. Biologists call these single base differences SNPs, which stands for single nucleotide polymorphisms. Researchers have discovered that certain sets of closely linked SNPs occur together time and time again. These collection of linked SNPs are called haplotypes, short for haploid genotypes. So to locate and identify many haplotypes in the human population as possible, the International HapMap project began in 2002. And the aim of the project is to give scientists a rapid way to identify haplotypes associated with various diseases and conditions, and to pave the way to more effective, life-saving medical care in the future. So copies of the human genome DNA sequence and those of other organisms are now freely available on the internet. Researchers and students can browse through the databases of human DNA and study its sequence. More data is added to these databases every single day. And one of the key research areas in the Human Genome Project was a new field of study called bioinformatics. Bioinformatics combines molecular biology with information science. It's critical to studying and understanding the human genome. So bioinformatics also launched a more specialized field of study known as genomics, the study of the whole genomes, including genes and their functions. So what have we learned? In June 2000, scientists announced that a working copy of the human genome was complete. The first details appeared in February 2001 in issues of the journals Nature and Science, and the full reference sequence was completed in April 2003, marking the end of the Human Genome Project 2 years ahead of the original schedule. So the Human Genome Project found that the human genome in its haploid form contains 3 billion nucleotide bases. Only about 2% of our genome encodes instructions for the synthesis of proteins, and many chromosomes contain large areas with very few genes. And as much as our genome is made up of DNA sequences from viruses and other genetic elements within human chromosomes. There will be a complete chart in your book that shows and compares the human genome with other organisms. More than 40% of our proteins are similar to proteins in organisms such as fruit flies, worms, and yeast. The Human Genome Project pinpointed genes and associated particular sequences in those genes with numerous diseases and disorders. It's also identified about 3 million locations where single-base DNA differences occur in humans, which may help us find DNA sequences associated with diabetes, cancer, and other health problems. I really cannot talk today. The Human Genome Project also transferred important new techniques to the private sector, including agriculture and medicine. The project catalyzed the U.S. biotechnology industry and fostered the development of new medical applications. The Human Genome Project worked to identify and address ethical, legal, and social issues surrounding the availability of the human genome data and its powerful new technologies. For example, who owns and controls genetic information? Is genetic privacy different than medical privacy? Who should have access to personal genetic information, and how will it be used? In May 2008, President George W. Bush signed into law the Genetic Information Non-Discrimination Act, which prohibits U.S. insurance companies and employers from discriminating on the basis of information derived from genetic tests. Other protective laws may soon follow. So the 1,000 Genomes Project, launched in 2008, will study the genomes of 1,000 people in an effort to produce a detailed catalog of human variation. Data from this project will be used in future studies of development and diseases, and it may lead to successful research on new drugs and therapies to save human lives and preserve health. So, in addition, many more sequencing projects are underway. An ever-growing database of information from microbial, animal, and plant genomes is expected. So perhaps the most important challenge that lies ahead is to understand how all the parts of cells, genes, proteins, and many other molecules work together to create complex living organisms.